Good morning, everyone. My name is Stephanie Murphy. I'm the director. I'm not going to do the acronyms first round. I'm the director of the Division of Comparative Medicine, DCM, within the Office of Research Infrastructure Programs, ORUP, in the Division of Program Coordination, Planning, and Strategic Initiatives, DPCIPSI, in the Office of the Director, OD, at the National Institutes of Health, NIH. So those are the important acronyms to know for today. First, I'd like to start off by acknowledging my colleagues, Drs. Miguel Contreras, Dr. Willie McCullough, and Dr. Siki Zhao, who have been the program leads from ORUP in developing and moving this workshop forward for today. I also want to acknowledge TJ Smith and Desiree Von Comer for their logistical expertise and assistance in managing presentations and the AV support for this workshop. I would also like to acknowledge the time and efforts of the organizing committee in putting together the agenda for the workshop today and engaging all of the speakers and participants who will be presenting with us. Last September, and many of you probably attended this, the NIH Office of Research Infrastructure Programs convened a workshop on zebrafish and other fish models, description of extrinsic environmental factors for rigorous experiments and reproducible results. That workshop brought together various stakeholders for plenary and breakout sessions on how to account for extrinsic environmental factors to improve rigor and reproducibility of experimental results using zebrafish and other fish models. Nutrition for zebrafish and other fish models was one of the factors identified at this workshop and is the focus of today's workshop. Currently, there is no consensus among aquatic facilities, researchers, and commercial vendors on what optimal reference diets should be for zebrafish during different developmental stages in order to minimize husbandry variation among aquatic laboratories and facilities. To address this gap, workshop organizers have put together several sessions to assess the needs and challenges of developing and implementing reference diets to improve animal husbandry and to promote more rigorous and transparent research in all areas of science involving zebrafish and potentially in other aquatic models. So in closing, I would like to thank all of you for your interest and your willingness to spend today being engaged in the session topics identified by the organizing committee and introduced by the speakers and panelists, as well as for the contributions you will all be making to the discussions throughout this workshop. It's now my great pleasure to introduce the chair of the organizing committee for this workshop, Dr. Stephen Watts from the University of Alabama at Birmingham, who will address the objectives for this workshop.
Perfect. Thank you, and thank you everyone for coming this morning. We'll go ahead and get started. We obviously are here for this workshop on defined diets and such. And I had put up here a picture of the zebrafish, indicating it's one of our target organisms that we wanted to be able to discuss today. In talking about other aquatic models, there are probably 25 other species out there that are used as models for biomedical research. Some of those are other fish, some of those are other invertebrates, but have been used in the role of research. And we hope to come up with some plans that we can help develop diets for other models as well. I had put on here some of these other aquatic models, and let me see if I can make it work here. Yes, the aquatic mouse right there, the aquatic pig, and of course we were using these models to study the aquatic nature of the humans. I obviously am jesting or facetious in this approach here, but the fact is all of these models, including the zebrafish, are incredibly important in what we're doing in trying to understand human nutrition, human disease, and human health for that matter. They share some, despite the fact that they're all very different, they share many similarities. For the most part, nutrition is quite similar among all of these animals. For a lot of people, you could look at the diets of these animals and find out that they're not that different, to tell you the truth. In addition, they also share diseases. These diseases that can be expressed in these various models are identical to what we see within the human structure. So it's important that we have these powerful tools that we can use to move forward in our investigations. I want to thank a number of people before we get started in making this happen. The NIH, for all the work that they've done. Miguel and his group have just done an outstanding job to get this thing in good stead. The organizing committee, a great job there. The speakers who came at a moment's notice to be able to come to this particular workshop. I must say that of every speaker I called to ask whether or not they'd participate, not a single one turned me down when they heard about the topic. It's very important to so many people that they said, sure, count me in, and on they came. So we really appreciate the speakers. Special thanks to the American Society of Nutrition Experimental Biology Ad Hoc Committee, who got together and are helping to promote this over the last couple of years to get to this particular point. And finally, personally, myself, I'd like to thank the Nutrition Obesity Research Center, particularly the one that helps to fund my work. We have one of the Zebrafish Nutrition Research Core there at UAB, and has allowed me to do the type of things that I enjoy doing. The organizing committee, Diana, Leela, John, Robert, Zoltan, we started meeting in the spring, and about every week there'd be a phone conference. Everybody participated to the best of their ability, did a wonderful job of coming up with ideas and the type of approaches we want to be able to use in developing this workshop. The NIH component with Miguel and all of his colleagues there just worked very hard to make sure this works out, or we produce a good workshop and hopefully some outcomes from that we will all enjoy. I want to remind us why we're here. NIH's mission, pretty simple: fundamental knowledge so that we can understand living systems, so we can enhance health. That's the charge of NIH. That charge has been there for well over 100 years, and the amount of work that has been done, the research has been accomplished, has made the NIH a powerhouse in terms of collecting information that can help us to understand human health. With all that we've learned, though, the complexity of the systems we're now starting to understand is far greater than we ever would have thought. And for that reason, we've had to increase what we believe to be the rigor and reproducibility in our work. Gone are some of the simple questions that we ask, because it's not that the systems have become more complex. To a certain extent, what we've learned now is that the systems are complex. We have a complex set of tools. We need to be able to look at this, but it requires a certain level of rigor to do it. And to maintain that rigor is going to be important for us to move science forward. Reproducibility is a major component of that. Together, these are the cornerstone of scientific advancement. In validation process that uses rigor and reproducibility, we still have some challenges. Statistics, I love the old days when t-tests would answer all my questions, but we're long since past that point. It's all multivariate and much more complex. That's what we have graduate students for. Reporting is something we all need to consider, because there's nothing more disappointing than reading a good paper and trying to reproduce that when in fact there's not enough information there. We need to understand that there's certain information we want to report and get better at it. We're pretty good, but we can get better. Transparency and data sharing, we need to all be able to work on what each other is working on, and that means we can't keep secrets from each other. We need to move forward, and to be fully transparent is how we're going to do it. One of the ways that this is going to work is through establishing guidelines or best management practices. And quite frankly, that's where we fall here now. We have a Zebrafish. It's a powerful tool. We need to make this tool even more powerful by understanding its nutrition. We can understand its nutrition by starting to develop diets that we can test, some of which will become ultimately, hopefully, reference diets. So we can develop best management practices for the zebrafish and for other models as well, so that we can move forward and maintain the type of rigor and reproducibility that is going to be required in our future work. Just a brief history from my perspective on this. The zebrafish became a model in the 1960s, and early on, what they did was feed whatever was available. That is, live feeds, some of which are still used today, off-the-shelf feeds. You go to Walmart, you pick up your feed, and there you went. I mean, that was fine. There was no problem with that. But we've gone past that. In 2010, a group got together at the Zebrafish Husbandry Association and brought forth a discussion. We need to do better. Let's develop potentially some reference diets in the future. And in 2012, myself, along with some of my colleagues, put together the first review on nutrition so that we can move in that direction. I remember being asked to participate in the review of nutrition, and I asked, how much space do I have? And they said, you have 20 pages. It took 1 page to write the review, because that's all the information, and 19 pages to say what we probably should consider doing. So we need to move past and beyond that now and generate additional information. 2014, we had a workshop. John Rawls had organized that at the Zebrafish meeting in Wisconsin. And we had a very good turnout to hear what we had to say about nutrition and zebrafish at that point in time. And I recall an investigator saying, nutrition is the elephant in the room. We know it's important, but we all really don't know exactly how to approach it. And so that's where we come to this particular point. 2 years ago at the Experimental Biology Conference, we assembled a group of interested individuals who wanted to promote perhaps the development of reference diets and our understanding nutrition. This group of individuals that you see up here is composed of NIH representatives, individuals, professors, and such. Several of the feed production groups were there. We had industry representatives such as DuPont and Dow had showed up. Quite an eclectic group of individuals who all were very strongly interested in promoting our understanding of zebrafish nutrition, and with zebrafish also other models as well. This group met for 2 hours, and you try to get any group together for 2 hours at an ad hoc meeting, and that's a difficult task. But everyone was interested. We moved forward, and we came up with some particular recommendations. Now, this obviously is a wordy slide, and I don't care that anybody reads much of any of it. But I wanted to put this up there because this is the identical page from the summary there. And some of these components are really important. We were going to address standardized diets. We need to move forward with an optimized standardized reference diet. We need to first involve experts from other diets to see what we can learn from those individuals. We need to start looking at databases that have information on animal nutrition so we can pull that together under a common umbrella. And that is exactly what this workshop is going to be doing. I recall there were 2 different needs identified. One is determine dietary requirements for different animal models. That's absolutely a need. And then improve our quality of reporting, which is going to enhance our rigor and reproducibility. So this is identical from 2 years ago, and I put it up there just to let you know that this is something that's not an afterthought. This is something that we've been developing for some period of time. Objectives of the workshop are as stated in your book. I'll just want to point out a few different ones there. We're going to review different diet development strategies in other model species as well to see how we can help ourselves within the zebrafish and other aquatic models. We're going to describe the need for these defined diets for maintenance and experimental stocks. We need to consider life stage, obviously. We looked at the potential impact of these reference diets on genetic stocks. There are so many different genetic stocks with so many different requirements. We need to assess nutrition and its value in using these or enhancing these genetic stocks that we've developed. A few other objectives. Number 5 to me is incredibly important. We need to identify obstacles and strategies and evaluate strategies that will lead to a successful consensus, acceptance, and implementation of defined reference diets. That is, we want to make sure everybody buys in and understands this is something that we need, and it's a discussion item that's important. We need to define an educational approach to informing the community and all of our associated partners. We have to educate ourselves on how we need to proceed. And finally, determine whether the approach to develop a defined reference diet could be applied to other aquatic models or animal models in general. The workshop program hopefully will do this. We've divided it into several different sessions. The first session is where we've come from, historical perspectives, and we're using a comparative analysis. We're going to look at rodents. We're going to look at Drosophila. We're going to look at fish production and evaluate these and see what we can use and borrow from these other models that will help us to define what we need to know and understand in zebrafish. That's where we've come from. Where we are at a working lunch, and we apologize for that. We had so much to cram into the one day that Zoltan will be leading a group here to talk about where we are and surveying our current status of zebrafish nutrition. This afternoon, the impact of diet variation on health and experimental outcomes. Zebrafish, we're going to show some of the research where nutrition is going to be very important for us to move this model forward. And finally, at the end of the day, and I know everyone will be tired, but this is probably one of the more important sections. We need to have an open discussion. We have some phenomenal minds here in this room that can contribute greatly to the approach we're going to need to be able to make to move this forward. So with that being said, I want one final request. As you watch the various talks today, think about them, and particularly you as speakers as well. If there are 1 or 2 or 3 major points that you think we need to keep up, we need to really think about, jot down a few notes on this. Because by the end of the day, if you're like me, I may forget half of what I think I'm going to say or talk about. But let's take a couple notes and see that we can understand what are the really relevant components we are learning from each of these talks that can be applied in moving this model forward by understanding more of its nutrition. So we're going to start there. In our first talk, I'll go ahead and introduce the first speaker, if that's all right. I met Forrest Nielsen. I met Forrest Nielsen a couple of years ago at the EB meeting. He goes by the name Frosty. He is the former director for the Human Nutrition Research Unit there at Grand Forks in the Dakotas, where it's nice and warm this time of year. Yeah, that's right. And he brings a unique perspective. The AIN 93 diets are the accepted diets currently used as reference diets for rodents. He was the chairman of that committee that made those decisions, building on what had come from the chair of the AIN 76 diets and taking us to that point. So he has an incredible amount of experience in diet development, and he was kind enough to volunteer his time to come and have a discussion with us, and perhaps we can learn about zebrafish and other aquatic model nutritional development by understanding what we've seen in other animals. So Frosty.